Welcome back, everyone, to another deep dive. Today, we're going deep into the world of metabolic and neurodegenerative encephalopathies.
Oh, those are a mouthful, aren't they?
They are, in dogs and cats specifically.
And they can be a real head scratcher.
Yeah, for sure.
Even for us neurologists sometimes, the clinical signs can be so similar.
Right.
And even on MRI, things can look really alike.
Exactly. And that is why this recent research from Frontiers in Veterinary Science caught my eye.
Ooh, good journal.
It is. It's titled Magnetic Resonance Imaging Pattern Recognition of Metabolic and Neurodegenerative Encephalopathies in Dogs and Cats.
I like the title.
It's a good one.
Straight to the point.
It really is.
Yeah.
Published in July 2024.
Okay.
And it dives deep into, like you said, they can look similar on MRI.
Yeah.
So how do we use those MRI patterns to better tell them apart?
Yeah, good luck.
Right.
Because, sometimes you look at an MRI and you're like, well, this could be...
Could be like 5 different things.
Yeah, it could be 5 different things. Which one is it?
Right.
So, the authors took on a pretty massive task.
Yeah.
They analyzed 100 cases, 81 dogs, 19 cats.
Wow.
With 15 different diagnoses.
15.
Confirmed diagnoses.
Gosh, okay. So before we get lost in the details, for our listeners, maybe just a quick overview.
Sure.
Like, what are we even talking about when we say encephalopathies?
Encephalopathy just means brain disease.
Okay.
Any disease of the brain. So, in this case, we're talking about encephalopathies that mess up the brain's normal function,
Ooh.
whether that's because of a metabolic problem,
Right.
like liver failure, or because of a neurodegenerative process, like a lysosomal storage disease where the brain cells are actually dying.
Got it. And so this paper is really all about using that MRI to distinguish between those two big categories.
Exactly. They're looking for patterns on MRI that can be like a fingerprint for each condition.
So you look at it and you're like, this is the fingerprint of
hepatic encephalopathy, or this is the fingerprint of, gangliosidosis.
Now, you mentioned that they included 15 different diagnoses.
Yes.
That's a lot. Can you give us a little rundown of what those were?
Sure. So on the metabolic side, they included hepatic encephalopathy, liver failure.
Yeah.
They included several intoxications.
Okay.
So like poisonings.
Got it.
And they included thiamine deficiency.
Okay.
Which can cause some pretty severe brain problems.
And you said this earlier, hypertensive encephalopathy.
Yeah, that one's a little bit controversial.
Yeah, why is that?
Because some people think it's more of a vascular problem, like a stroke, rather than a metabolic problem.
Interesting.
But, the authors of this paper included it in this category.
Okay.
So they're kind of lumping it in with the metabolic encephalopathies.
All right. So they're pushing those boundaries a little bit of what we think of.
They're broadening the definition a little.
So on the neurodegenerative side, you said there's a bunch of them.
Yeah, there's a whole bunch. So they included some lysosomal storage diseases.
Okay. Like
neuronal ceroid lipofuscinosis, or an NCL, gangliosidosis, and fucosidosis.
Wow. It sounds like a genetics lecture.
Right.
I'm bringing back flashbacks from vet school.
I know, right?
Okay, so lysosomal storage diseases.
And then they also included L2-hydroxyglutaric aciduria.
I cannot wait to say that one all episode.
Yeah, that's a mouthful.
Okay, so L2-hydroxyglutaric aciduria.
Lafora disease, spongiform leukoencephalomyelopathy.
Okay.
And cerebellar cortical degeneration.
Wow. Okay, so they covered all their bases.
Yeah, they tried to be as comprehensive as possible.
It sounds like it.
Both common conditions and some really rare ones.
Seems they didn't just rely on the MRI, right?
No, they didn't. They actually confirmed all the diagnoses with additional testing.
Okay.
So like blood work, genetic testing, and even histopathology in some cases.
So looking at the brain under a microscope.
Exactly.
So we know for sure that these MRI findings are tied to these specific diseases.
Exactly.
That's a real strength of this paper.
It is because, in a lot of studies, you don't have that definitive diagnosis.
Right. You're just kind of
you're just kind of guessing.
Assuming.
Yeah.
Based on what you're seeing.
But in this study, they really went the extra mile.
Awesome. Okay, so let's get into some of the nitty-gritty.
Okay.
The cool stuff.
Yeah, the interesting findings.
What are some of the big surprises they found?
Because there were quite a few surprises. Some things that, we didn't really expect based on our previous understanding of these conditions.
Oh, this is where it gets good.
Yeah.
Okay, so spill the beans. What'd they find?
Okay, so let's start with hepatic encephalopathy.
Liver failure.
Liver failure, right.
Yep.
So classically,
Yeah.
what we've been taught and what we see in other species is that you see a bright signal on T1-weighted images in a certain part of the brain called the lentiform nuclei.
Okay.
But in this study, none of the dogs or cats with hepatic encephalopathy showed that pattern.
Ooh, interesting.
It just wasn't there.
Okay, so the classic sign was missing?
It was missing.
What did they see instead?
They actually found involvement of the cerebellar nuclei.
The cerebellum.
The cerebellum, which is involved in coordinating movement.
Right, so like smooth
movement, balance.
Yeah.
And that's a completely new observation.
No.
It's never been reported before in dogs or cats with hepatic encephalopathy.
So does that mean that the clinical signs might be different in dogs and cats?
That's the million-dollar question. We don't know.
Oh, man.
It could be that this finding has just been overlooked in the past.
Okay.
Or it could be something unique to dogs and cats.
Wow.
So definitely something that needs more research.
Fascinating.
So another interesting finding was in the dogs with myelinolysis.
Oh.
They all had the expected involvement of the thalamus, but they also had consistent involvement of the superior longitudinal fasciculus.
Okay, now hold on. Break that down for me a little bit.
Okay, so the superior longitudinal fasciculus.
Yeah. What is that?
It's this big white matter tract that connects different parts of the cerebral cortex.
So like a pathway.
Yeah, like a superhighway of information flow in the brain.
Okay, so if that's damaged, then communication is messed up.
It's messed up, yeah.
Wow.
And we've never really seen that before in dogs with myelinolysis.
So this means that maybe myelinolysis affects a wider area.
It might, yeah.
Than we previously thought. And it raises the question of why.
Yeah.
Why is this particular tract so vulnerable?
Why that one?
Why that one? Could be something about its metabolic demands or the types of cells that are in there.
Right.
But we need more research to figure that out.
That's so interesting. Okay, so we got liver failure, we got myelinolysis. What else is there? Well, thiamine deficiency had an interesting finding, too.
Yeah.
They saw involvement of the oculomotor nuclei.
Oculomotor.
Oculomotor, yeah.
What does that even control?
The oculomotor nuclei control eye movements.
Okay, so eye movement problems.
Yeah.
We don't typically think of that with thiamine deficiency.
Right, exactly. It's not something we typically see clinically.
Okay.
But this study suggests that it might be more common than we realize.
Maybe we're just not looking for it.
Maybe. Or maybe it's just really subtle.
Right.
And we just haven't noticed it before.
Oh, fascinating.
Yeah.
Okay, so what about some of the neurodegenerative diseases?
Well, hypertensive encephalopathy had a pretty striking species difference.
Okay, so high blood pressure in the brain.
Yeah, really high blood pressure.
Okay.
So both dogs and cats with this condition showed changes in the white matter.
Okay.
But in cats, the changes were way more extensive.
Oh, wow.
Yeah, they had more widespread white matter involvement.
So way worse in cats.
Yeah, much worse.
Interesting.
They also had mass effect. Which means that the brain was swelling and pushing on other structures.
And that can cause all sorts of problems, right?
It can cause neurological signs and even death in some cases.
Wow. So cats.
Yeah.
Not handling that high blood pressure as well.
It seems like they're much more sensitive to it.
Why do you think that is? What's the difference between dogs and cats there?
Well, one possibility is that cats have a different kind of blood vessel arrangement in their brain compared to dogs. And this might make them more susceptible to the effects of high blood pressure.
Okay.
So their blood vessels might be more fragile or more prone to leaking when the pressure gets too high.
Okay, so moving on to those inherited neurodegenerative diseases.
Right.
Any big surprises there?
Neuronal ceroid lipofuscinosis.
NCL,
yeah.
Yep.
Showed severe brain atrophy.
Okay, so shrinkage of the brain.
Shrinkage, yeah. Which is what we would expect.
Right.
But two of the dogs also had this pachymeningeal enhancement.
Okay, pachymeningeal, hold on.
I know, it's a mouthful.
That's a big word.
It basically means that the outer layer of the brain, the dura mater, was highlighted on the MRI after they gave contrast.
So that's not normal.
Not normal, no.
What does that even mean?
It could mean that there's some kind of inflammation going on in the dura.
Okay.
Maybe even an autoimmune process.
Interesting.
And that's not something we typically see in humans with NCL.
Okay, so another, possible species difference there.
Yeah, possibly.
Huh.
So that's something that needs further investigation.
Definitely. Okay, so then what about gangliosidosis and fucosidosis?
Those two conditions showed similar MRI findings.
Okay.
They both had increased signal intensity in the white matter.
Okay, so that's kind of like a common theme we're seeing here.
Yeah, it is.
White matter involvement.
White matter involvement.
But this time, it's not because of a toxin or myelinolysis.
It's because of this genetic metabolic disorder.
Right. Okay, so lots of different things can cause white matter changes.
Lots of different things.
Makes it tricky
It does make it tricky.
to figure out what's going on.
Yeah, you really have to look at the whole picture.
Okay, so then what about some of the rarer conditions?
Yeah. Like L2-hydroxyglutaric aciduria.
L2.
I know, it's a mouthful.
Okay, L2-hydroxyglutaric aciduria, what'd they find with that one?
All the dogs with that condition showed a really consistent pattern.
Right.
They had this thickened and hyper-intense cortical gray matter.
Okay, now remind me, what's the cortex again?
The cortex is the outer layer of the brain.
Okay,
It's where a lot of the higher level thinking happens.
got it. Okay, so the cortex is thickening.
Yeah, and it's hyper-intense.
Okay.
Which means it's brighter on the MRI.
Brighter means bad, usually.
Usually, yeah.
Okay.
And they saw this in both the cerebrum and the cerebellum.
So, like, all over the brain?
Yeah, pretty much.
Wow. Okay, so that's pretty distinctive. It is, yeah.
Okay, so then what about Lafora disease?
That one was a little bit more challenging.
Okay.
Some of the dogs had normal brain morphology.
So, like, their brains looked totally normal.
Totally normal. But others had mild atrophy.
Okay, so a little bit of shrinkage.
A little bit, yeah.
But not a lot.
Not a lot.
So why the difference?
Well, it could be age.
Right.
You know, older dogs tend to have some degree of brain atrophy anyway.
Yeah, just like people.
Exactly.
Okay, makes sense.
So it's hard to say for sure whether the atrophy they saw was due to the disease or just normal aging.
Okay, so it's kind of a confounding factor there.
Yeah, exactly.
Okay, so then spongiform leukoencephalomyelopathy.
Yeah, that
That one's a mouthful.
That was a mouthful, and they only had one case in this study.
Oh, wow. Okay.
It was a young Belgian Malinois,
Okay.
and it showed lesions in the cerebellar nuclei.
Okay, so cerebellum again.
Cerebellum again, yeah.
What was up with that dog's cerebellum?
We don't know for sure.
One case? Hard to tell. Hard to tell with just one case.
Yeah.
But it's interesting because that finding is actually consistent with what we see on histopathology.
Okay, so when they look at the brain under the microscope,
Yeah.
they see similar changes.
Similar changes, yes.
So even though it's just one case, it kind of supports
It kind of supports what we already know
what we think we know.
about this disease.
Okay, but you did say earlier that cats can be different.
Oh, yeah. Cats are always different.
Why?
They like to keep us on our toes.
So with this one, how are cats different?
Well, in cats, the MRI findings can be much more variable.
Okay.
They can have lesions in the white matter, like we saw in the dog,
Okay.
but they can also have lesions predominantly in the gray matter.
Which is the opposite of what we usually see.
Yeah, it's kind of the opposite.
Wow. Okay, so cats, once again, throwing us a curveball.
So where's the curveball?
Okay, so then finally, cerebellar cortical degeneration.
Yeah, this one's a classic.
Makes sense. Cerebellar cortical degeneration.
Yeah, the name kind of says it all.
It does. So what did they see?
All the dogs and cats with this condition showed cerebellar atrophy.
Okay, so shrinkage of the cerebellum.
Shrinkage, yep.
Okay.
But there was a difference between the dogs and the cats.
Oh, here we go again.
Here we go again.
Okay, what's the
But
difference?
atrophy was much more pronounced in the dogs.
So worse in dogs.
Yeah, worse in the dogs.
Okay, interesting.
Again, it could be an age thing.
Right.
The cats in this study were younger than the dogs.
Okay.
Or it could be that the disease just progresses more slowly in cats.
Okay.
We don't know for sure.
So cats are just, like, more resilient.
It seems like they might be.
Huh. Interesting.
But, you know, more research is needed to confirm that.
Of course. Always more research.
Always.
Okay, well, that was a whirlwind tour of all these different conditions.
Yeah, we covered a lot of ground.
We did. So before we wrap up this part, I kind of want to step back
Okay.
and look at the big picture a little bit. What are some of the, like, overarching themes that emerged from this research?
Well, I think one of the biggest takeaways is that MRI is a really powerful tool
Yeah.
for evaluating brain disease in dogs and cats.
Right. It can show us things that we just wouldn't be able to see otherwise.
Exactly.
But it's not a magic bullet.
No, it's
You
not.
can't just look at an MRI and say, oh, I know exactly what's going on.
Right. You have to interpret the findings in the context of the whole clinical picture.
So the history,
The history,
a neuro exam,
the neuro exam, yeah.
blood work,
Other tests.
yeah, all of that.
All of it.
It all fits together.
It's like a puzzle. You have to put all the pieces together.
And sometimes you're still missing a piece.
Yeah, sometimes you are.
But this study gives us some more pieces
It does.
to work with.
Yeah, it gives us a better understanding of what those pieces look like.
Right, and how they fit together.
And how they fit together.
Okay, so MRI is powerful, but
Yeah.
it's not a magic bullet. What else? What other big takeaways?
I think another important point is that there's still so much we don't know
Oh, yeah.
about these conditions.
The brain is a mystery.
It is. It's incredibly complex.
Yeah.
And we're only just beginning to scratch the surface
Right.
of understanding how these diseases work.
So more research is always needed,
Always.
which I'm sure is music to the ears of all the researchers out there.
I hope so.
Okay, so we don't know everything, but we're learning more all the time.
We are.
What else? What other big picture takeaways?
I think this study really highlights the importance of pattern recognition.
Oh, yeah, you mentioned that earlier. Like recognizing those subtle but distinctive,
Yeah, the subtle clues
those,
that can point us toward the right diagnosis.
that's like detective work.
It is. It's very much like detective work.
Except the clues are in the brain
Yeah,
and the suspects are diseases.
exactly.
Wow, I like that.
So we have to be trained to see those clues,
Right.
to recognize the patterns.
And it takes time.
But it does. It takes time and experience.
So, for our listeners who are maybe newer to neurology,
Yeah.
don't get discouraged.
Don't get discouraged, no.
It's a learning process.
It is, and it's a lifelong learning process.
It's always changing.
Always changing, yeah.
Okay, so pattern recognition, really important.
Very important.
What else? Big picture.
I think the species differences are really interesting.
Oh, yeah.
We saw that with the hypertensive encephalopathy.
Right, cats are different.
Cats are different.
And with the cerebellar cortical degeneration.
Yeah, and with several other conditions.
Right.
So it just reminds us that we can't always extrapolate,
Meaning?
meaning we can't always assume
Okay.
that what we see in one species
Is going to be the same.
is going to be the same in another species.
Got it. Yeah. Dogs and cats are different,
They are.
even though we lump them together sometimes.
We do.
They're very different creatures.
They are. They have different physiologies,
Right.
different metabolism.
Different everything.
Pretty much.
So we have to be mindful of that
We do.
when we're diagnosing and treating these conditions.
Absolutely.
Okay, well, I think that's a good place to pause for now.
Okay.
We've covered a lot of ground,
Well, you have.
and I'm sure our listeners' brains are full.
I hope so.
But we're not done yet.
No, we're not.
We have a lot more to discuss.
We do.
So stay tuned for part 3 of this deep dive.
Where we'll be talking about
We'll be diving even deeper
even deeper.
into the world of metabolic and neurodegenerative encephalopathies.
Can't wait. So, picking up where we left off, are you ready to delve back into those complex brains?
Oh, absolutely. I'm fascinated by all these new insights, especially those unexpected findings in hepatic encephalopathy.
Yeah, the involvement of the cerebellar nuclei, that was a real curveball.
It really was. It makes you wonder if maybe we've been missing something all along.
It's possible. Sometimes we get so focused on the classic signs that we overlook other possibilities.
Right, like we're so used to seeing those T1 hyperintensities in the lentiform nuclei that we don't even think to look elsewhere.
Exactly. And this study reminds us to keep an open mind, to be willing to challenge those long-held assumptions.
To think outside the brain, so to speak.
I like that.
Okay, so cerebellar nuclei in hepatic encephalopathy. Big surprise. What else stood out to you?
Well, I thought the species differences were particularly intriguing.
Yeah, like with hypertensive encephalopathy, how the cats were so much worse off than the dogs.
Yeah, it's almost like their brains are wired differently.
Right. Like they're just more sensitive to those high blood pressure spikes.
It seems that way, and that just underscores the importance of understanding those species-specific variations.
Totally. We can't treat a cat like a small dog, or vice versa.
Exactly. We have to tailor our approach to the individual patient, taking into account their species, breed, age, all of that.
Speaking of tailoring our approach, I was really impressed by that flow chart they developed, the one for MRI pattern recognition.
Oh yeah, figure 14, that's a really practical tool.
It is. It kind of simplifies things, breaks it down into those key features, brain volume and lesion distribution.
Right. It helps you narrow down the possibilities, kind of like a decision tree.
Exactly. So if you see increased brain volume, you might be dealing with something like an intoxication or L2-HGA.
Or feline hypertensive encephalopathy.
Right. And if you see isolated cerebellar atrophy, well, that pretty much points you toward cerebellar cortical degeneration.
It does. Now, of course, it's not a foolproof system.
Of course not. You always have to consider the whole picture.
Always. But it's a great starting point, especially for those who are maybe less familiar with neuroimaging.
It can help you avoid going down those rabbit holes, chasing after diagnoses that don't really fit.
Exactly. It helps you focus your diagnostic workup.
Okay, so we've talked about the surprises, the species differences, the pattern recognition tool. Anything else you want to highlight before we wrap up?
I think just the overall message that there's still so much we don't know about these conditions.
Oh, absolutely. The brain is a complex beast.
It is. And we're constantly learning new things.
This study just kind of peeled back another layer of the onion, so to speak.
It did. And it opened up all sorts of new avenues for research.
Right, like why are those cerebellar nuclei involved in hepatic encephalopathy? What's the mechanism there?
Yeah, and why are cats so much more susceptible to hypertensive encephalopathy?
And what about those pachymeningeal enhancements in NCL? Is there really an autoimmune component?
So many questions. And that's what makes this field so exciting.
It does. It's like we're on the edge of some major breakthroughs.
We are, and I can't wait to see what the future holds.
Me neither. Well, this has been an incredible deep dive. Thank you so much for sharing your expertise with us.
It was my pleasure.
From Vet Neurojar, keep those minds inspired, hearts light, and tails wagging.